Well, welcome to Lab Week 7 of Bio 225. So, for this week, we are going to be discussing several different biochemical tests, and then there are several virtual biochemical labs that I do want you to complete, as well as a few kind of YouTube videos to look at procedures of the tests that we're going to talk about today. So the first test is a catalase test. A catalase test is used to see if the bacteria have an enzyme known as catalase. Now, we did talk about the catalase enzyme briefly in chapter 6. The catalase enzyme is an enzyme that can break down hydrogen peroxide, which is what we see here, into water and then oxygen. So we talked about this enzyme in chapter 6 when we talked about aerobic and anaerobic bacteria. So if you remember, and that slide's actually this slide here, aerobic bacteria are able to utilize oxygen because they have enzymes to kind of break down oxygen into usable forms. Whereas anaerobic bacteria do not have these enzymes, so they typically tend to stay away from oxygen. So if you remember, when an aerobic bacteria takes in oxygen, that oxygen initially becomes toxic, and it becomes something called a superoxide ion. But that's okay for an aerobic bacteria because aerobic bacteria have an enzyme called superoxide dismutase that can break down the toxic superoxide ion into hydrogen peroxide and then normal oxygen. So then the kind of second step is, since bacterial cells don't want hydrogen peroxide in their cells, they have to take an enzyme called catalase, which is the one we're talking about. And catalase is an enzyme that will break down that hydrogen peroxide into water and then just normal oxygen that the cell can use. So typically, if bacteria have these enzymes, that means that they can use oxygen and they're aerobic. Whereas if a bacteria did not have these enzymes, that means they're anaerobic and they don't use oxygen. So really, when we do a catalase test, what we're testing to see is if our bacteria are able to utilize oxygen or not. If they have the catalase enzyme, then yes, they can. If they don't, that means that they do not have the ability to utilize oxygen, and they're most likely anaerobic. So the procedure is terribly easy, and you will see this when you go to do the virtual catalase test that I have in D2L. All you have to do is take a clean microscope slide, put a few drops of just regular hydrogen peroxide, it's just the same hydrogen peroxide that you probably have in your house, and to those 2 drops of hydrogen peroxide, you would add, it doesn't have to be 2 drops, however many samples you're doing, but to that drop of hydrogen peroxide, you would add a sample of bacteria. So just drop of hydrogen peroxide, add sample of bacteria, then all you have to do is watch to see if it bubbles or not. If it bubbles, that is a positive result. That means that the bacteria that you added has the catalase enzyme. It's breaking down that hydrogen peroxide into water and oxygen, and that's what's causing the bubbling. And your bacteria is probably aerobic and prefers to use oxygen. However, if you add bacteria to the hydrogen peroxide and it does not bubble, that's a negative reaction. That means that your bacteria does not have the catalase enzyme. Your bacteria is not able to break down hydrogen peroxide into water and oxygen, and your bacteria is most likely an anaerobic bacteria. Now, we've all used hydrogen peroxide before. I mean, I remember being younger and you'd fall and get a scrape, and that would be like the first thing my mom would do, would dump a bunch of hydrogen peroxide in the wound and it would burn like crazy, but it was kind of cool because you saw all this bubbling. And you think, and I thought this even, up until early years of college, you think that that bubbling is like the bacteria exploding and dying. But unfortunately, that's not what is really happening. When you put hydrogen peroxide on a wound, the bubbling that you see, some of the bubbling that you see, is actually the bacteria that are present breaking down that hydrogen peroxide into water and oxygen because they have the catalase enzyme. So the hydrogen peroxide isn't really killing all the bacteria. If the bacteria that's in the wound has the catalase enzyme, you're not going to kill them with hydrogen peroxide. They're going to be able to break down the catalase enzyme into water and oxygen. So for that reason, hydrogen peroxide is not the best at killing the bacteria. So when you get a cut or a wound, you really don't want to use it alone. It's good to use initially because the bubbling, while it doesn't necessarily kill all the bacteria, that bubbling kind of dislodges any dirt and debris to help clean out the wound. But in order to really effectively kill the bacteria, you want to follow it up with something like alcohol that's going to be much better at killing bacteria, and then obviously some Neosporin to provide that kind of antibiotic relief for the bacteria as well. But just know, if bacteria have the catalase enzyme, you're not going to kill them with hydrogen peroxide. Typically, it's best to use something like alcohol to really efficiently kill bacteria. So another test that you'll be doing a virtual lab on is the oxidase test. So the oxidase test is used to see if a bacterium has this enzyme called cytochrome C oxidase. Basically, if your bacterium has an enzyme called cytochrome C oxidase, that's a good indication that they're aerobic and they prefer to use oxygen. So it's kind of the same principle. We're trying to find the same thing to see if our bacteria use oxygen or not. However, if the bacterium does not have the cytochrome C oxidase enzyme, that's a good indication that it's anaerobic. So this test tells us if bacteria use oxygen or not as well. It's just doing it in a different way, looking for a different enzyme. Instead of looking for the catalase enzyme, we're looking for the cytochrome C oxidase enzyme, which typically we'll know if it's present or not because our test will turn colors. As we can see here, a positive test will turn this kind of bluish purple color, whereas a negative test will be clear. So when you do an oxidase test, you are given this thing called a dry slide. And this dry slide, it kind of looks like a little window, but it has the oxidase reagent kind of built into these little paper filters that we see here on the dry slide. It's so simple, and you'll see this in the virtual lab. All you have to do is take a sample of bacteria. So you would just take a sterile cotton swab, collect a sample of bacteria from your petri dish, and just smear it onto a little portion of this dry slide. And then you just wait 20 seconds to see what happens. If it turns blue within 20 seconds, like what we see over here with B, that's a positive reaction. That means that yes, your bacteria has the cytochrome C oxidase enzyme and it is probably aerobic. However, if it stays clear the whole 20 seconds, that's a negative reaction. That means your bacteria does not have the cytochrome C oxidase enzyme and it is most likely anaerobic. This test does have a time limit. It has to turn blue within 20 seconds. If it turns blue after 20 seconds, let's say it turns blue at, 25 seconds, that's not technically a positive result. It could be a false positive. What happens is moisture from the air can sometimes affect the test and cause it to turn positive when it shouldn't. But that typically doesn't happen until after 20 seconds. So that's why you only have that 20-second window to read the results. But if it turns blue within 20 seconds, that is definitely a positive result. So the next test is called a SIM test. The SIM test is nice because it actually does 3 things at once. SIM actually does stand for something. It stands for sulfur reduction, indole production, and motility. So the nice thing about this test is it's only one test, but it shows you 3 different things. It shows you if your bacteria can reduce sulfur, if they're able to produce this substance called indole, which we'll talk about, and if they're able to move or not. So the procedure for this test is a bit unique, and I do want you to watch the video in D2L to see how the procedure is done. But basically, you're given this test tube of sim media, which it's a solid media inside of a test tube. And you actually use something called an inoculating needle to do something called a stab inoculation. So it's very similar to the inoculating loops that you've been seeing in the PowerPoints. It's just that instead of having a loop at the end, it's just straight like a needle. So all you would do is you would collect a sample of bacteria from your petri dish, and you would take that inoculating needle and stab it right into that test tube where there's media, and then bring it straight back out. So you go straight in and then straight out. And then you would incubate the test tube for a period of time, 24 to 48 hours. And then after that period of time, you would look at the test tube to interpret your results. One of the results requires that we add this reagent called Kovacs reagent to see if a color change happens, and I'll show you that in a second. But to really get your head around it, watch the video in D2L, and it actually shows someone going through the procedure just so you can kind of see how that was done. So, like I said, this test is a bit complex because it does show you 3 different things. So, let's say we do our procedure, we stab inoculate bacteria into the test tube, we incubate for 24 to 48 hours, then we look at our results. So, if we look at our test tube and it turned black, if it turned black, that's an indication that sulfur was reduced. So, in these 2 samples, this one right here, sulfur was reduced. That's why we know that, because we see how it turned black. But in this one over here, this bacteria that was stab inoculated into this test tube was not able to reduce sulfur. So that's a negative reaction for sulfur reduction, whereas this one over here that turned black is a positive reaction for sulfur reduction. Then we'll kind of jump to this one over here with mobility. With mobility, which is movement, what you want to do is look at the stab line, because when we stab inoculated these test tubes, we took that sample of bacteria on that needle and we kind of put it straight in and straight out, and that produced this stab line. So after incubation for 24 to 48 hours, and you look at that stab line, you need to look to see if it's thick or if it's thin. So a thick stab line, like this one here, indicates that the bacteria are able to move, which means they probably have flagella. Because when we stab them down in here, over that 24 to 48 hours that they were incubating, they would have started to swim sideways. And that's what would have created this kind of thick, hazy line. But after 24 to 48 hours, if you look at your stab line and it's still very thin, like what we see over here, that's a good indication that the bacteria are not able to move and don't have flagella. So when you stab inoculated them and you placed them wherever they landed, they just stayed right there and grew from that spot, but they didn't move outwards. So to kind of assess mobility or movement, you just have to look at the stab line. If it's thick, they're able to move, they have flagella. If it's thin, they're not able to move, they do not have flagella.